Okay. So we start with polygenic traits in sort of semi-random order. So I'm going to talk in other lectures about mutations, about gene mapping, about other things. So this is about statistical genetics of complex phenotypes and about polygenic prediction. Okay, so I'll start on a philosophical note. Why in the world we're doing genetics, right? So that's the first question you have to ask yourself. And what we think life science is, there is some genotype, and it gives rise to some functional biology, some complex clockwork machinery that we don't really understand very well. And that clockwork machinery produces phenotypes. Right? So that's how we think about life sciences generally. What genetics tries to do, genetics try to bypass this clockwork machinery and link genotype to phenotype directly. That's a very weird choice, right? Because for many of us, we're really interested in this functionality. Why in the world we're doing this? So there are three answers, at least as far as I'm concerned. So answer number one is evolution, because evolution happens by changing frequencies of genotypes, and natural selection affects phenotypes. So if you want to study evolutionary dynamics, this whole clockwork machinery is in principle unnecessary. Second part is medicine, right? So usually when a patient comes to a doctor, so the patient doesn't say, you know, my protein-protein interaction isn't tight enough, so my two proteins don't interact well enough. Or like, you know what, my gene expression is off. Like I'm uncomfortable with my gene expression. That's not what's happening, right? Usually patients complain about phenotypes. And again, we can study risk of developing certain phenotypes. We can study predisposition to certain phenotypes by linking genotype to phenotype. And the most important third part of this is that if you look at the biology textbook, genetics provides the window into this complex clockwork machinery. Because the easiest way, whatever you concern with, some biological phenomenon, disease, right, effect, the easiest way to find the window into the machinery is to link to genes. Right? That's what we do. This is why we use genetics. Okay, very basic. Okay, guys, how many of you took statistical genetics class or like, you just let me know if something is boring, right? So the next few slides will be introduction. So generally the idea is that we can think of genetics as something Mendelian or simple. Classic experiment on P by Mendel, segregation of disease in human pedigrees. I'll be talking in my public lecture later today about this. However, the issue is that if I look at real populations, like fish in the pond, I don't know, students on this campus, and I measure whatever property, binary or quantitative, it doesn't behave like this, right? So if it has some statistical laws, but they are not laws of Mendelian genetics. And that's what led over 100 years ago now to this Mendel's by nutrition model. Okay, guys, sorry, I'll be checking temperature in the audience. How many people know about this stuff? Okay, about half. So I'll go more than half. Okay, so I'll go real quick. Okay, so you know there was Mendel's by nutrition model, so this was a serious debate, and Mendelian genetics was considered to be an oddity. And the idea was that we have these real laws. And in this real laws, this is one of the examples of statistical laws of genetics. So people are lined up, and height is normally distributed. Height is pretty inconvenient phenotype, but it's used always as very obvious one. So I'll be keeping using it as an example. Now, what's happening with this? I always thought that this is some sort of an approximation. That, oh, everything's approximately normal, height's approximately normal. We'll look at thousands of individuals from first healthy visit patients in Mount Sinai Hospital System in New York. It's dead normal. Like all high moments are indistinguishable from zeros. It's just unbelievably normal. And people look at the UK Biobank and 500,000 people, and it's normal. I have a couple of friends who said like, oh, low normal fits maybe a little better. With this numbers, low normal is pretty much normal. So it's absolutely normal. It's unbelievably normal. So that's, by the way, how many parameters does normal distribution have? Do we know? 2. That's right? So because we're interested in variation, so the mean is not really relevant here. All the science is about science about variance. Okay, and this is a paper, 1877, which not shown on the slide. This is work by Dalton, single author paper in Nature, and that's very curious. So this is after Mendel's work, but this is before Mendel's work has been translated to English, right? So Dalton didn't know apparently about Mendel's work. So he selected P for his experiment, which is curiosity, but he looked at the size of the P pod, not the shape or color. And I didn't know about this work. I actually stole this from one of the slides by Nick Barton. And what he says that in the cross of P, right, so progeny of smaller plants are smaller, progeny of larger plants are larger, but variance is identical, which is a very non-trivial observation. And he says he has no explanation for that, but it simplifies speculations a lot, so he accepts that as convenience. And this big Dalton plot, again, you probably know that. So this is height of children as function of mid-parent height. And what we see in this distribution, I use it for teaching, we see a few things, right? So we see that children of taller parents are taller than, are on average tall, children of shorter parents on average short. We see that children of taller parents are not as tall as their parents, and children of shorter parents are not as short as their parents, and this is what Dalton called regression to mediocrity, and this is why we think that a lot of this is genetic, but not all of that is genetic. And very curious observation is that if you do regression lines here, both regression lines are linear. Right? And the variance, conditional variance is the same for any phenotype. Right? So conditional variance is constant. Which actually means that the distribution is bivariate normal. All the conditionals are normal, and all the marginals are normal. Guys, are you with me? Am I going too fast? Everybody's okay. Am I going too slow? No. Okay. Okay. I receive my positive reinforcement. I keep going as I've been going. Okay. All the marginals are normal, or all the conditionals are normal, and it's a bivariate normal distribution with the density written on the slide. Now this you probably know, and that led to infinitesimal model. And infinitesimal model now is hobby horse in the human genetics, even though it's old. I don't know who invented the term. But the original idea of infinitesimal model, if I have a pedigree, human family, right, or animal cross or plant cross, joint distribution of quantitative trait for the pedigree is given by a multivariate normal distribution, and the pedigree defines the covariance matrix. Right? So it's not only that if I line up everybody, the distribution is normal, or parents and children are normal, but I can take whatever pedigree I want, and number of meiosis will determine my covariance structure, and it still is going to be normal. That's probably not exactly true. It's very hard to believe it's exactly true, but a lot of modern statistical genetics uses it in a very different formulation, because now we look at DNA, and I'll talk about that. I don't remember if I'm talking about specific mathematical aspects of that, but again, I'm happy to jump and discuss that. Okay, so again, I should go probably quickly because you know all of this. Fisher paper 1919, right? So this was a resolution of Mendelian bionutrition debate. You have the phenotype, and you assume that there are quantitative trait loci. So every locus pushes it a little bit up, down, a little bit up. And every locus, I have no idea how he guessed it, right? So now it's very obvious to us because we know about polymorphism in DNA, and that's a clearly discrete object. He didn't, but he postulated that this is a Mendelian trait. And a bunch of those, together with non-genetic influences, produce this normal distribution. That's the model we use today. That's the important model. We use it for binary traits as well, usually in the form of liability threshold. If we have time, I'll talk a lot about that at the end. So the idea here that if I have a binary trait, then I assume that there is unobserved, normally distributed quantitative trait, and the chance of the probability to develop binary trait is just the tail probability here, right? This falls over the threshold. That's not the only model, but that's a very popular model. It's called liability threshold model. And generally everything is the same. So now, the question is, from here, what's the map between genotype and phenotype, right? How linear it is, and this is what I'm not going to talk about unless there will be questions. We can talk whether the map is linear, whether there are interactions and stuff like that. The other question is how many loci, how multidimensional is the whole situation? Do we have any guess? Do we know? Guys, do you know how many loci underlie different traits, or we should go slower? Maybe I keep talking about history, and then we'll come to the section. So the early indication that the model is relatively polygenic comes from selection experiments, right? And this is Illinois long-term selection experiment. Again, how many people know about this? Okay, good. Then I'll explain it. This is not type of experiment a single graduate student does, right? Because the data here are from 1896 to 2008. It's over 100 years worth of data. This particular line is the oil concentration line. So maize is not very oily plant. So you take corn, it's 5% oil concentration, and you start selecting for increased oil content. And there is a linear response for 100 years. If you go down, there is a response down. I mean, it's hard to hit zero, but it also looks like linearish response going down. You can change direction from high to low. It's very plastic and it responds. So this model is this result is absolutely inconsistent with 1, 2, 3, 4 loci determining oil content in maize. Why is that? Because very quickly you would fix this variance, right? So you would have, if there are 3 loci, there will be a jump, another jump, and total saturation, right? So you cannot produce constant linear response. Constant linear response may exist in 2 different ways. In a little bit more mathematical sense, the change in mean is proportional to genetic variance, and this is what is called price theorem. We can discuss it. I wasn't going to talk about that. But change in responses is proportional to basically covariance between fitness and phenotypes, but this is we're selecting on the phenotypes, so it's primarily proportional to variance. So it means that genetic variance is determined by a very large number of loci, or maybe you're picking up new mutations as you go. Again, meaning that a lot of loci are involved. I really love to talk about this. Again, I wanted to go faster, but I'll probably spend a little bit of time here. Maize is a long generation time system. It's inconvenient to do. Probably talk about public initiative a little bit about this as well. So this is the experiment on Drosophila. Drosophila is very quick. So it's very hard for me to believe in this. I read the original paper, the review, people say it's correct. So this is selecting Drosophila for speed of flight. So the experiment is set up in the following way. So you take flies, Drosophila flies, right? And do you know what flies eat? Genetics group. They eat yeast, right? So yeast lives in fruit. You take fruit, you put the fruit, you release them, and they fly towards the fruit, and they fly like 2 centimeters per second roughly. Like if you imagine a fly, this is how a fly flies. Then you select the fastest one, breed them, get their progeny, and introduce a fan, blowing wind against the direction of the flight. Right, now they have to bottle the flight, right? They bottle the wind. You take those who bottle the wind successfully, breed them, select next generation. It's a linear response, and you end up with Drosophila flying almost 2 meters per second. I cannot imagine these flies, right? It's hard to believe they exist. I don't understand why Pentagon is not like the Department of Defense doesn't fund flight genetics. I mean, that's what they should be doing, right? So again, it's the same story again and again and again. It's a very plastic response, and the lack of saturation here and the lack of obvious jumps suggests that there are many loci. The question is how many? Is it 10? Is it 50? Is it 100? We don't know from this data. Now GWAS came, I assume people know roughly what GWAS is. If not, I can quickly explain.
It's the first time I've heard it.
Okay. Then I should step back. I have another slide deck. We will discuss this very carefully. Most people are here for tomorrow, right? I'll be talking about that tomorrow as method for gene mapping, but I briefly will explain it to you. So what we're doing right now, we have the ability to sequence the genome or to do microarray genotyping. Microarray genotyping, it means that we sequence a bunch of people and we find, say, 2 million variable position SNPs, and we put them under the microarray. So this means that I can go into a very large population, for example, the UK Biobank, which is now a big force in the community, and I know variation at every locus in each individual. Then conceptually, it's very easy. I can regress height, for example, on genotype on this locus. Or if I have people with diabetes and no diabetes, I can say, okay, which allele is more common? I can draw logistic regression and say which allele is more common in diabetics versus non-diabetics. Then there are gazillion statistical details that make it very hard. But in this simple conceptual framework, it's that simple. Right, so what I'm doing an association test as opposed to linkage test, right? So I'm not testing for segregation or co-segregation. I'm testing just for the allele being present in individuals with phenotype versus with no phenotype. And I'm finding statistically significant associations. And the whole business started in 2007. And this is from my friend Chris Katsapas about multiple sclerosis, how the field progressed. They had this many SNPs they used to type. In 2007, they found 2. And this was about 1,000 people. They found 2 signals. This is, I believe, these are non-HLA signals from multiple sclerosis. And then they found 1 more with more SNPs and pretty much doubling the size. And then they increased number of SNPs and again increased sample size, and there are 6. And there are 3 new. With going to 6,000 people, you go to 10,000 people, you get 25 new hits. 27 new hits, 100 new hits you get after meta-analysis, and then you're very quickly in thousands. Right? So what it means that signals are so weak, right, that I cannot find them with the regression analysis on thousands of individuals. And there are probably so many. Now we have the first iteration GWAS, so genome-wide association study. It was just published in Nature in height. It's called saturation because they explain very large fraction of genetic variants, but it doesn't mean they found all the loci. Right? There are probably more of weak effects. And there are 12,000, and probably the total number is maybe 3 times that, I would say, right? Because as soon as like effects that are measurably non-zero, it's a complex statistical inference, confounded by a lot of other signals. So the question is like, do we really believe in all this inference? But I think by now the community believes in an extraordinary polygenic model. So this is from a paper by Luca Conner. It's the method based on Fourier transform for the data. So I will explain roughly what it is. So this is number of loci he infers. This is inference. This is not direct measurement, right? So this is a complex statistical inference. But this is number of loci. Guys, it's clear that, right, that I'm using the term locus, which is genetical non-committal term, right? I'm not saying regulatory origin, I'm not saying gene. I'm using the term locus as pretty much independent signal in space of linkage of equilibrium. So what you see here is that median number of loci for height, right? So this is number of loci with effect more than median, is predicted is in, this is 10,000, so this is a few thousand, so they're probably a little bit over above median now with 12,000, so somewhere here, and it will go up. But what's important here, the other side of this, because these variants are very common, these are not very rare variants in the population. That's what is interesting. These are common variants. They are very frequent in people. So this means that if we have many of them, the individual effect size is very small. So the effect size, median effect size, is 1 over 10,000 of standard deviation, and standard deviation height is 5 centimeters, roughly. You're in centimeters here, right? So you're going to say a couple inch, but I know I should say 5 centimeters. So you divide by 10,000 and you're in small fractions of a millimeter. So each of these genetic effects increases or decreases height by a small fraction of a millimeter. Very small fraction, right? Maybe hundreds of a millimeter, tenth of a millimeter. So this is body mass index, right? Another classic anthropometric measurement. This is risk of schizophrenia. Again, almost 10,000 loci at the median. So it should be, obviously below the median we have less than above the median, right? So there are probably a few dozen thousand of loci. And this is inflammatory bowel disease, which is a little less polygenic, about say 1,000 loci. But again, the story is repeating itself again and again. It's a very large number of loci with extraordinarily small effect sizes. It's fundamentally different from Mendelian genetics. I'll be talking about Mendelian genetics later in the public lecture, and especially tomorrow. But this is a very different story, right? It's not one gene large effect, it's tens of thousands of loci. Now what I wanted to do, since we sort of understood GWAS, I'm at the end of my introduction. There will be introduction to more specific things, but I'm at the end of the global introduction, so we're in this polygenic world. And I want to talk today about two problems. One problem is, can I predict disease risk for anybody in this situation? Because if my effects are minuscule, if my effects are small fractions of 1% of increase in risk, what can I do? And I also wanted to talk about evolutionary genetics, about the forces that maintain this variation erect. Right? Are we sort of clear about the introduction? I can move. Good. Okay, I don't know how many people still know the movie Gattaca. Many do. It's a Cheetah slide. If you talk about genetic prediction of traits, you have to show the slide. I'm sorry, I know everybody's bored by this, but I have to do it again and again because this is how you're supposed to start. That's it, okay? Okay, so what is happening? A genotype at every locus, you measure genotype at every locus, it has a minuscule amount of information, right? So the early idea is that we'll find a gene for diabetes, I predict diabetes looking at the gene for diabetes, none of that works, literally none. But it does not mean that I cannot predict the phenotype. Now, the idea is that I can measure heritability, and if you know what the Dalton plot, you probably know heritability of height is 80%, right? And the way because we're in this kind of world of normal distributions, we measure everything as fractions of variance. And one other thing, which is absolutely unbelievable, people are still working on this. It seems that for complex traits, for small effects, genotype to phenotype map is dead linear. Very hard to believe. I'm happy to discuss that. I've had an interesting experience. You still remember there was such a thing as COVID, right? So before COVID, you go give seminars in many places, sometimes invited by graduate students. You go for dinners or lunch with students, and at least one student would say, "Oh, now we have hundreds of thousands of individuals, and my project is measuring nonlinear effects, and I'm measuring, and I found nonlinear effects, and these are interesting nonlinear effects." And I'm like, "Wow." And then COVID hits and you don't travel anywhere for 3 years. And I'm like, where are the papers? Where are these papers from the students who found them? Nobody found anything, literally, right? Still debated, but at the first approximation, the whole map is linear, so as soon as you're in, Mendelian genetics is not linear. But as soon as you're in small effect sizes, genetics are like calculus. You're in the linear map. So this means that if I build a linear predictor, because my heritability is measured as a linear effect of genes, my accuracy of my predictor could be as good as, ideally, as heritability. So this means that if heritability of early onset heart disease, early onset myocardial infarction is 50%, most of the diseases around 50, 30, 50, 60, I can do actually pretty well. I can do better than most clinical indicators do. So it's not a hopeless predisposition. Now, I'll talk about practicality first, then about history, and then about methods, including one we published recently. So this is LDL cholesterol, and this is risk And LDL cholesterol in this study measured like 30 years before the outcome. And this is prevalence of calcifications, arterial calcifications 30 years past. So what you see is that the risk goes linear with cholesterol much earlier in life. Now, what is happening here is this. So why are we worried about predicting risk practically? It's a non-trivial question, right? So do I worry about predicting risk of blood pressure? Well, I don't know. I mean, as soon as your blood pressure systolic is below 120, definitely 117, there is no improvement in risk. So I can say, okay, I have a treatment option. I have a measurement. If your blood pressure is 140, I give you medicine. If you below 120, you're happy. Here, we have statins. We have drugs to drop cholesterol. Everybody benefits, right? It's a linear relationship. There is no obvious subgroup of the population who would benefit from therapeutics. The only way to select it, well, there are a couple ideas, right? So one idea is, let's put statins in tap water. So I'll just put drugs in the drinking water, everybody would consume them, and people would be happy, right? So somehow the population is not ready for that, for multiple reasons, right? We wouldn't discuss this. Like, I see people, we have companies, well, you're recording this, I shouldn't say so, okay? but there are people who want to genetically re-engineer liver, and amazing people, amazing results. So I'm like, do I want to genetically re-engineer my liver to keep my cholesterol low? It's another thing, right? But the only thing you want to know risk. That's the only reason you drive the risk low. Okay, and genetic risk is very good because you can estimate long-term risk at birth, you can combine it with biomarkers, and you can get a non-trivial data. So, the whole story about polygenic prediction actually started before any DNA sequencing. And it started in large pedigrees, as many things in genetics, and this is Charles Anderson. And these are papers in the 1970s, based on an infinitesimal model. I'm looking at the clock. I think we don't have time to talk about that carefully. I don't know how many people know what random effect models are. Okay, let me try. How many people know what blood is? Good. Okay, so we have one person exactly. Okay, I suggest I skip it for now. We have the discussion section. We can talk about that. So that's. But the basic idea is this. So we discussed this logic that if you have a pedigree, you have a multivariate normal distribution for the pedigree. Now you can imagine, because they work with cattle, there was herd of the state of New York. That was the first application. You have enormous pedigrees. So you have very big covariance metrics. And this was, I think, the first application of linear algebra in genetics. So they started doing prediction for cattle. And now, of course, this moved to DNA sequencing and actual prediction based on genetic markers. In application to humans, in 2007, Peter Vischer and Ray and Mike Goddard, so they moved from cattle genetics to human genetics, and they said, oh, there are so many interesting studies. Why can't we predict the same way as people predict in cattle from multiple genes, right? Again, this is a non-trivial idea. This is the idea that I don't measure like a Mendelian clinic. I don't measure allele and I predict based on the allele. I'm building the overall statistical predictor based on everything. And they said, oh, this probably wouldn't work because it's too small of the effect and it's too little data. And then they were part of the schizophrenia and bipolar consortium with Tom Sklar and Sean Purcell in Boston. And at least the way Peter tells the story, he saw the kind of blip of the effect in the, he used to be in Edinburgh in Scotland, blip of the effect in the Scottish court. So he emailed to Boston to Sean, and Sean ran the analysis. And what they did something very simple. They took independent variants, proving LD. So they said, okay, if two variants are correlated, I just exclude one. So I keep only non-correlated pairs. And I exclude all effects that are really very small, probably zero. And it's suddenly significantly non-zero. The signal was very small, but it was clearly non-zero signal. Then everybody got very interested in this. A bunch of people extended ideas of blood, going to Bayesian. There's a very interesting method BSLMM, which goes into loss in one limit and in the random effect model in the other, and so forth. And then what happens in the community? So again, what's happening is this. So we work with data, and the data are association data. And association data, you can get in two ways. You can get direct raw data on individuals. So this is every individual's genotype, this is every individual's phenotype. It's hard to compute, it's a lot of computation, because data sets are massive. Also, people weren't willing to share, right? So now we're kind of beyond the transition point with initiatives like the UK Biobank. We have data available for everybody. But it used to be that people would run the study, and what they would share is summary statistics. And they would share regression coefficients, they would not share the raw data. So there was a lot of interest in developing methods that are based on summaries that don't require raw data. Bjarne Williamson with Alkes Price, they developed this method. Yeah, so what's, I don't know if it's included. You can get to infinitesimal model very easily with the following assumption. That is, imagine that every locus in the genome has the effect. We don't know which, but we draw it randomly from normal distribution. In that case, it's very difficult to beat a very simple sum of apparent effects. However, in truth, we have very many loci. We have 10, 20,000 loci, but it's still a minor fraction of the genome. So one way to do it in a Bayesian framework is to say, okay, so I have this betas. Betas are my coefficients, the effect on genotype and phenotype and locus I, and I would draw them from normal distribution with the variance, which is the number of markers, so this is heritability, or with zero. So this model is called spike and slap. It's a delta function at zero in the normal distribution, right? So this is why it's spike and slap. So it's either zero effect or some effect. And you can build a Bayesian framework around it. So, okay, so this became popular, and at some point, Seka Teresa and Amit Kerry came up with this paper, which is very interesting, because all of that was kind of statistical genetics playground. And these guys are clinicians, right? And they said, look, if you're in the clinic, we have cases of familial hypercholesterolemia, very high cholesterol and very high risk of heart disease, which is a Mendelian mutation, segregating in the family. We have treatment guidelines. We can use our treatment guidelines for in this case. However, if I run polygenic risk, I can identify individuals of very high risk, and there will be more of them. Why can't we apply the same treatment guidelines to this group? And this made the whole field super interesting for everybody, because suddenly it's practically applicable. And whether it will be or not, it's still a question. I think the balance is shifting towards probably yes. But we will still have to resolve a few things. And one thing we have to resolve, this is paper by Alicia Martin, and I can talk a little more about that. It's not transferable between congenital populations. So the current machineries are not transferable between populations. It really depends on your ancestry. And by the way, the same thing it happens in agricultural species, right? So you go from one breed to another, you can transfer. And what Alicia, I'll talk about that. Again, I don't know, I'm watching the clock. I'll try, if I go fast enough, we'll talk more about this. But the idea in this paper is this. The biology seems to be absolutely the same, right? So same loci, same effect sizes, but you run through different demography, different demographic models, right? So some people stay in Africa and population grows, some people go through bottlenecks, through migration, and what happens is correlations between loci change and frequencies change a little bit. And because in this study, we have no zero-zip idea which individual variants are biologically significant. We work with highly correlated statistical structure. These changes in correlations completely ruin our ability to learn in one population and transfer the learning to another one. And that's the greatest bottleneck, right? Especially in the United States, where population is very heterogeneous. It's very difficult to apply this in the clinic, right? Okay, Guys, is this okay? I'm watching the time. I can either go quickly through basic stuff, or at this point I wanted to switch to something a little more technical and walk you through one of the papers we had recently, and then maybe a little more about the scores, or I can go quicker and skip details. Are people interested in mathematical details a little bit? Okay, I know yes. I assume people who didn't say no, they also yes, because they didn't say that. Okay, so look, this is a very simple idea, right? So I have a phenotype that I measure in everybody. I call it Y, and I have a locus effect beta. And this is my genotype, which is 0 dosage of risk allele 1 or 2, right? Because we're diploids, 2 copies of each chromosome. Now, and there will be kind of extra stuff here, but extra stuff is not genetic. So what I have to do, I have to do this. So why wouldn't I estimate this better and multiply by genotype, and I can predict all genetic effect idea? So the reason, of course, is that I have no knowledge of betas. I only know beta S, and this is what we use in stats, right, to say that this is not the true effect, it's an estimated effect. And there are two issues that are complex. One issue is that I have sampling error, and sampling error, if my effects are close to 0, sampling error would propagate into beta hat. That's a non-trivial thing, because you think about regression as an unbiased estimator, right? So you're thinking, oh, if I have beta hat, an expectation it's a beta. But I'm giving you another thing. I'm saying that I have a whole bunch of effects, right? And many of them are 0. And I'm asking what's the beta hat conditional on true beta. That's a different question. So this noise would propagate here, and I would count a lot of noise in my predictor. So one possibility is what to do. I can just cut the noise away and say I'm only taking larger effect variance. And I can say, okay, so I just produce weight for each variant as function of estimated effect size. And if I cut some variance to 0, this is how my function looks like. It's probably not ideal function. It's somewhat okay, but it's not ideal. And this is p-value thresholding. I can do Bayesian, so I can assume now watch this, because this is complex, right? So we just said that it's expected beta hat given beta is beta. I'm giving you the object expected beta given beta hat. In frequency statistics it doesn't exist, right? So beta is a parameter, it doesn't have expectation. But I'm Bayesian, so I assume that there is such a thing as expected beta. And this gives me this function that I'm reweighting every snap, given this expectation. Now, in addition to noise, there are two other things. So one is linkages equilibrium. And linkages equilibrium means that my variants are correlated along the sequence. And my true effect at locus 1 induces the effect at locus 2, even if it's indeed 0. And the effect at locus 2 induces the effect at locus 1. Right? But that's not all the story. The very early methods already corrected for that. Noise is correlated, because if I sample haplotypes, right, so I sample groups of variants, not only my expectation, but my variance on the sampling of frequency is correlated between the loci, and that's an issue. So what we developed is, again, we'll talk about other methods. I'm just using one we developed because it's easy for me to talk. This is what some tuned primarily in Statistical. It's called non-parametric shrinkage, and the idea is that we estimate it without a prior, and the other thing is we base it on D. Now I'll do this. I'll talk first about the method like LD doesn't exist, because otherwise it's hard, and then I'll talk about how we treat LD. So what we do, we take all SNPs and group them according to betas, so we put them in buckets. And every bucket has a score like this, right? So we have additive polygenic score for SNPs in each bucket. This means that I took the median parameters and reduced my parameter space to like 10. And this also means that I took piecewise linear approximation to this curve I'm trying to estimate. Okay, guys, are you with me so far? Yeah. Now, and I can use my very small number of cases and controls to learn 10 parameters and produce the score which is optimal. Now the problem is there is LD, And if there is LD, I cannot do this trick, right? Because this guy, I need more time. That's unfair. I need more time. Oh, am I actually out with the term of time? 5:15. Okay. Well, I'll try to be relatively quick. Thank you. So this guy is correlated to this guy, right? So I cannot use Vikings because I would put the same signal. So what we do is this is our LD, right? So there are two SNPs B and A, and I have correlated LD structure. I have correlated frequencies. So I can find a projection, which is the standard eigenvalue decomposition. And then what happens is I just put people in a different basis. Now, this does not work with the full matrix, so we have to do windows, and there are a lot of serious issues with regularization and other things that I'm not going to talk about. But the basic idea is that as soon as I do this orthogonalizing transformation, so now every individual doesn't get allelic dosage, but gets a linear combination of allelic dosages. And what I didn't realize, it doesn't only decorrelate allelic effects, but it also decorrelates noise. So we can show that this decorrelates noise, so that works quite well. Now the problem is now my effect sizes are not normally distributed, as God knows what kind of distribution, and I cannot bin even my, we call them eigenloci, because each comes with eigenvalues. So now suddenly I don't have 10 parameters but 100. Anyway, so this is how we do. These are data in the UK Biobank, so small parameter training is within the UK Biobank. And some of those are, I think this curve is training in the UK Biobank and moving to Mass General Brigham data bank in Boston. So bottom line is, at the 5 percentile, you can get, depending on the disease, breast cancer, inflammatory bowel disease, type 2 diabetes, coronary artery disease, you get 3 to 4-fold increase in risk. It's a relative risk. I'm not a clinician, it's hard for me to say whether breast cancer risk increase in 3-fold is sufficient to send the woman to a mammogram or not, but there are, of course, strategies and people are considering this quite seriously. Very briefly, I'm almost out of time, I'll go through other methods. So PRSCS is the method working quite well in our hands, and this is Bayesian method, but it's with a very flexible prior, so it allows you a lot of different shapes, right? Not only spike and slab, and that works quite well. There's bias R, where you have multiple variance scales, and you can now suddenly use functional information about SNPs. And use them in your mapping. So again, it's a Bayesian method. There are methods that are extensions of lasso, if you're familiar with that, right? So this is more regularization fixed effect technique rather than Bayesian. LDAC Bolt works again pretty well, and the idea is that unlike tools that we have now, it uses varying prior based on allele frequency and functional effects. And we ran for critical assessment of genome interpretation. There is this kind of assessment of different methods. So Sonia and I ran this challenge, and what Sun found is this method worked very well compared to a lot of others. Well, not a lot, some others would assess. Okay, this is the set of methods. Surprisingly, you can still gain something out of mathematics, because at some point I thought that who cares, right? You just get more samples, and more sophisticated method like payoff is very minimal, but it doesn't seem so. Now, what makes polygenic CRISPR non-transferable? One is, of course, all human populations in common variation share the same variation, right? But frequencies are slightly different, and this is how we determine ancestry based on genetic data. And as soon as your frequencies are different, you change your parameters in prediction, and especially differences in liquid disequilibrium, so difference in covariance structure. I can think of this too as differences in variance covariance metrics for genotypes, right? And that's what's super important. The question is if this is the only way. Now, how to get out of this problem, because if this is the only issue, I can do better biology. I can find out which variants are actually functionally significant, or do fine mapping, find variants and build predictor based on biologically significant variants, and I will resolve the problem of transferability. The other one is to think about very better algebraic solution, which so far nobody came up with. The other one, it's probably minor contribution, is differences in actual effect sizes. I was recently shown data in Africa where people with very similar ancestors live in completely different environments and they have different effect sizes. I was part of this work by Hohem and August Price. We looked at genome-wide association studies between East Asia and the US. So you look at primarily European population and East Asian population, and genetic effects are highly correlated, but they are not one. They're like 90%, 85, 90%. And the only reason we think that's possible is gene by environment interaction. Right? So there are a lot of effects of environment. So you know what's the biggest genetic risk of lung cancer, right? So if you run a genetic study, the locus is nicotine receptor. Because the biggest risk of lung cancer genetically is how much do you want to smoke. That's environment specific, right? So if you go to the population where there's no smoking, there's no genetic effect. So this is what G by group is. I think that's probably minor effect. So there is this method PolyPred by Omer Weisbrot, who tries to use this approach, what I just explained, trying to use functional data, not trying to find truly biological signal. We'll talk about that tomorrow, about how to find truly biological signals. We'll talk much more biology later today and tomorrow than statistics. And this is probably more transferable. Okay, I have a fun story about evolutionary forces, but watching the clock, I'm out of time. So I guess you will not learn that. Okay, let me do one. Can I take like 2 minutes? I'll just give a gist.
Please take as much as you want. Please take.
Are you sure? Okay, this is actually very new work. It's still unpublished. It's you're recording it, right? I didn't realize that. That work is still unpublished. We're trying to finalize it, but I'm very enthusiastic about this. So the question is, why do we have? So we discussed two things. We discussed how polygenic is the variation. We discussed how we use it to predict traits. Now the question is why in the world does it exist? What's the generative model in terms of evolution? And think about this. So when I look at rare disease, right? Genetic disease is rare. It's eliminated in the population very quickly by natural selection. Now suddenly, we have common variants, right, involved in disease like schizophrenia or diabetes, and those things are actually bad for you, and epidemiologically bad for prevention. So natural selection should eliminate all of this disease. Why doesn't it? And this is kind of a surprising thing. So the originally, my original idea was this, that clearly all large effects, the reason we don't observe them, the reason we observe thousands of the small effects is because natural selection eliminated all the large effects. And in population genetics, there is this idea of, we call it drift barrier. So the situation where stochastic rearrangement of alleles, which we study in diffusion theory, is a stronger force than deterministic force acting like natural selection, right? So you just cannot fight noise at some point. Natural selection cannot fight noise. And I used to think that the standard model is these things are bad for you, but natural selection cannot prevent this very small effects arising and they accumulate. Turns out that's not the case. It's at least not in this simple form. Sorry, this is like the epidemiological point and this is there was a lot of papers saying that natural selection actually does exist in this population. So I should speed up and not discuss this. So this is the observation. The observation was made by Eric Lander, was shown to us. We call this smile plots. And I think this is quite stunning. So look what's happening. This is blood pressure, this is schizophrenia. Right? Now, this is frequency of risk. Now what's happening is, in the middle, you never have variance of large effects. There are no common variants. So variant that 50% of individual carries never has large effect. Why we don't have those? We think because of natural selection, right? Because they are bad for you, natural selection gets rid of them. Now there are variants where very few people have, right? And those have large effect on blood pressure and schizophrenia. So those are diseases on top of those are quantitative traits. That's on the aldosterone. And that's because of some reasons. Some reasons are entropic reasons. It's actually not immediately obvious what gives the curve. But one reason is natural selection pushes alleles towards rare. Rare alleles might bring large effect. Also, there are many more rare variants than common variants, so there is kind of an entropic type effect, right? So there are just more variants. And there is power, right? Because here I have no statistical power to learn anything because they are rare and small effect. I have no statistical tools to find anything in this region. But what's absolutely surprising to me, right, is that the symmetry. Because the fact that there are 1% of individuals in the population who carry a variant with large effect on blood pressure or schizophrenia that cannot exist in the middle, I'm okay. But 99% of individuals also carry variants increasing the risk that cannot survive in the middle. So that's kind of weird. Okay, I know I was given extra time, but I would not exploit generality. Oh gosh, I don't know why. This is probably moving between the computers. These are results of the simulations. These are computer simulations, and there should be like a lot of dots here. I cannot show this, but okay, I can try to kind of explain where the dots are. Right? So if there is no selection, there are still a little bit more things on the it's symmetric, obviously. There are still a little bit more things on the sides because there are no variants on the power. But it's pretty flat, so you don't really need a lot of smiliness here. There's still a lot of stuff around in the middle, right? If natural selection doesn't happen. If it's directional is the model what I believe would be happening. Mutations are bad for you, they are eliminated. And the intuition is that it should be all one side. But that's not true because you might have fixation of a deleterious variant. You might have a back mutation which is beneficial for you. And that creates bind shape, but it's always skewed. And stabilizing selection, I always like talking about stabilizing selection when I teach a class about that because stabilizing selection means that I prefer average phenotype. And I'm exactly average height, I'm average cholesterol, I'm average like everything, except HDL. Like what in my medical report, HDL is the only thing that is not average, everything else is average, so I like the idea of stabilizing selection. That prefers, and this creates pretty smiley situation. Obviously, right, because of stabilizing selection, if I prefer to be in the middle, right, both sides are bad, the effects are larger, these effects are suppressed. And then we also looked at the model, in this model from a paper by Yuval Simons and Gaisela and others, and they are saying that it's not like we measure one trait, we're actually measuring a lot of traits. And you can think about a geometrical model like this, thinking that you're not in the optimum in one dimension, but you're in the optimum in a very highly dimensional sphere. And that induces a specific distribution that has very nice properties on this particular model. So we tested that, and that's pretty close to one dimensional. It has, if your properties, it generates a little high effect in the middle sometimes, but also with the bulk of the data following this power line. Okay, so mathematics works the following way. So we have to do two things. We have to map allele frequencies to selection coefficients. Selection coefficients is yellow dose of fitness. And what's written here is deterministic part of allele frequency change. There is also stochastic part. Right, so it's not a stochastic equation. This is just basically a simple form of deterministic part. And there is connection between selection coefficient and effect on the trait. And this really different for different types of selection. And then the model is formulated in terms of base factors. And what's happening, to our surprise, the directional model actually never wins. It's always a stabilizing model, and stabilizing model primarily pleiotropy has high support, although this is very unstable inference, so we're a little bit worried about presenting this. And what's interesting, if I look at height or BMI, I have massive bias factors in favor of stabilizing selection. I'm not really surprised, right? These are quantitative traits. If I look at disease traits like type 2 diabetes and schizophrenia, I'm very surprised because I think those things are negative. And in this liability threshold model, it seems that average liability is preferred. And I don't know what it means. It's a little bit easy to think about for diseases like hypertension or hypercholesterolemia, where liability is literally a physically measurable object. And sometimes in type 2 diabetes, because glucose level is physically measurable object, right? It's a measurable thing. But turns out that we see that stabilizing selection maintains these diseases in the population. I find it super surprising. So this is, so look at 15 phenotypes, and yeah, except one big exception in our hands, and this contradicts some of the literature. We had one cancer phenotype in the data set with sufficient power, and that's breast cancer, and that comes up as then neutral, so we don't see any selection acting on breast cancer lesions. I'm a little bit puzzled by this result, but it's very stable. That base factor is very stable, 0. So don't worry. Okay, again, a lot of this was work in this lecture was not our original work, and this is more meant to be teaching part, but for the original work, people in the lab, Evan Koch, Noel Connery, I think I didn't include the slides at the end, but Jonathan Mitchell is another grad student. Our collaborators at the Broad Institute, Alex Blumendahl, Nick By now at Oxford in the UK, Ben Neal and Eric Lander, as I said, made the first observation, and other collaborators both in the lab and outside the lab. And since I'm on the acknowledgement slide, this is the current lab, and this is some of the pictures of the lab, and this is Cape Cod, about an hour drive from where we work. Okay? Thank you very much. Sorry I'm over time. Are we doing questions? We don't do questions. Whatever. I don't know what's the format. Should I jump to the whiteboard? Should we go to enjoy coffee and do questions by like whatever you want, guys?
Can you redefine the stabilizing selection thing?
Yeah, okay. Do this since I should redefine because all my slides were bold, so it's hard to say, right? So I have the effect size. We usually call it beta because of regression coefficient, okay? And I have allele frequency, and allele frequency I can measure in multiple different ways. I can measure non-reference allele, I can measure minor allele, I can measure derived allele, I measure risk allele. So if I have a locus, right, there is this allele and this allele, I'm saying this increases the risk. Its frequency of risk increases. So it goes not from 0 to 50%, but it goes from 0 to 1. Okay? Okay. Now, imagine what's happening for a rare disease, monogenic disease. Mutation happens. It's strongly deleterious mutation. It has very high selection coefficient, reduces figures, meaning that allele frequency cannot become high, right? So when you're very rare, you can still maybe fluctuate stochastically because your variance is, your stochastic variance is pretty large compared to selection, but you can never become a common variable, right?
Yes.
So what do you expect? You expect that your effect size will be pretty big, you'll sit here, and you will never be here or here. Now I have phenotype with small effects. And the first thing I have to draw here is a power curve. So that's purely defined by power. So, concept of statistical power is reasonably familiar to everybody, right? Or it's not? Yes or no?
Yes.
Yes. Okay. I take it as a yes. If you say no, we'll go into this. If you don't say no, I assume it's a yes. So the power means obviously if my allele is rare, I have 100,000 individuals, but I only have 3 people with this variant, right? There is no chance I will detect significant effect on my trait. So if I'm rare, my minimal effect size should be here to have any power to detect the effect. If I'm common, I'm very well powered, right? So this goes down. And this leaves roughly on the scale of x1 minus x, right? So that's the variance, where x is the frequency. So this is my envelope. So things here just don't exist for me. So now I'm looking at common disease. Now I'm saying, okay, the effect size cannot be that high. But if I'm rare, my effect size may be here, here, right? Maybe also here, 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 but here I have no knowledge. Now, I'm going up in frequency. So this is, say, 1%. 1% of chromosomes, or say 1% of, roughly, of individuals are heterozygote for this variant. Now I move to, say, 50%. At 50%, these guys will not propagate to 50%. The selection will not let them, right? And generally, few things propagate to 50%, even by a random drift, but generally, those selected one would. But I would still have small ones, right? Because the small ones, with very small effects, these effects are so small that selection is inefficient. That's not surprising. What's surprising is I have these guys here. What's surprising is that I have exact symmetry. Now, what the symmetry means? Symmetry means, if my selection is directional, naively, I can say, okay, so I spend some time here. We call them population genetic sojourn time, right? So I spend some time here in this frequency bin. Here, it's almost deterministic movement. But if I manage to come across, I also spend some time here. Or it may mean that mutations are coming from this side. That somehow we as humans fix too many diabetes predisposing alleles and then mutate the other direction, right? Now the problem is, for this to have balance, by the way, one non-trivial, it's relatively simple mathematics, but it's a non-trivial intuitively. If I have 50% alleles, now this is not frequency, this is how many alleles I push from this direction. And 50% alleles that I push from this direction, so 50% alleles are deleterious, bad for you, right? And 50% alleles are beneficial, they are protective against disease. I reproduce exactly the neutral curve. So my curve would be like this. So my points would be much higher in this middle region. So I know this is not what's happening. If I keep balance, I always produce some asymmetric. It will be still U-shaped. It will be kind of smiley shaped, but it will be crooked, right? It will be non-balanced. Now, what we try to fit, what we fit very well is the classic model of stabilizing selection, meaning that, so you usually say, okay, so I have a trait, blood pressure, don't right? So fitness is frequently modeled as exponential function, is exponential square function, right? So it would be something like of this sort, right? Sorry, this is phenotype, this is fitness. So this is not good, and here fitness is great. I can move it to a very multidimensional space. It's very natural model for quantitative traits. But why is we fit exactly the same model for diabetes and schizophrenia? I find very surprising. So it seems that genetic liability for a bunch of diseases is maintained by stabilizing selection rather than by directional selection. It's a new result. I don't have any good biological intuition about it.
Thank you.
Okay. Any more
Okay.
questions, or everybody needs coffee?
Now we can take a break, and continue.
Okay.